Well, for something now entirely different. Maybe not. So which is the click? Oh, there, this one. Yep. When I first came to Purdue 6 years ago from MIT, I started going out and meeting with various faculty, deans, department heads, and asking, what kind of problems do you have? And their response was, we don't know what to do with the data that's being generated by all the faculty, by the researchers. And I said, oh, we can help with that. And I get this quizzical look from people saying, how? And I said, well, that's what we do, is we organize things. We put structure. And it started occurring to me that as a librarian, we had been very bad about telling people what it is we know and what it is we do, as opposed to what people seeing us do in the libraries. A library is an organization, is a structure of information, of content. We've happened to be dealing with, in the past, with tangible objects. That same premise, the same organizational structure, can also apply to digital data. And the other question I would get, or comment that I would get from faculty, when I would say, we can help you, they would say, oh, that's great. And then there would be this other kind of fear in their voice. And they said, but yes, librarians like to share everything. We don't. And I said, well, okay, I think we can work with that too. And archival science is a different aspect. Library science and archival science are not the same. Archival science allows there to be controls applied. So when somebody gives a collection to archives, they can put restrictions, they can put embargoes on that collection being seen or used. And they also have a process where they can do selection, where they can do sampling of an archive and not keep the entire archive. Whereas librarians, we have a very difficult time throwing anything away. So as I just said, this is really consistent with what we are doing in the libraries and organizing information. When I spoke to Jim Bottom, who was the CIO at the time when I arrived here at Purdue, and asked him what his biggest problem was, and he said, it's managing all of the data that faculty are generating. And I said, oh, we can help. And he looked at me and he said, I feel like the cavalry have arrived. And I said, we have. Well, I was naive. And then I would hear comments from librarians saying, we don't deal with the research data. We only deal with the end product, the final article that's published, and we preserve and maintain that for the future. And I thought about it, and I said, no, that's not true. We deal in raw bits of data already. In archives, you think about all the manuscripts, all of the data that are used by humanists and by social scientists. Those are raw bits of data. They happen to be tangible because they're print. But until a researcher uses them and turns them into a research project to answer a hypothesis, they are still just raw bits of data. So how has the research process changed since the advent of computational science from bench science? Either I'm going forward or I've just duplicated that page. Oh, I know what. I forgot that I have to hit the button about 6 or 7 times to get that one to come up. In here, what we're showing is the relationship of data to the process that raw data moves through, from being collected through sensors or through experiments into processed data sets, or analyzed data when a researcher has actually looked at the data, come to an hypothesis, published the data, and then possibly through a preprint into the published research in a non-traditional format, possibly in a digital repository, or published research in the more traditional sense. But what we're also looking at is how do we make the data set itself a citable object. Obviously, we know that we can make a research article citable, and that citation has an impact upon what a person's reputation may be in their field, how they may proceed through promotion and tenure. But why shouldn't a data set that has a significant impact in the field not be also recognized? So we are working at the Purdue Libraries. The Purdue Libraries, along with the California Digital Library, which is the central system for the University of California, are working on an international collaboration that is developing and assigning DOIs to data sets, the digital object identifiers. Those will then be linked to such as articles that are published, so that when a person publishes an article, there's a DOI assigned to that. That's already being done. There are hundreds of thousands of DOIs being assigned so far for research articles. The underlying data is not being cited. This is what the DOI is to do for the data sets. We just had a meeting in London last week where the International Consortium, which is representing most of Western Europe, Australia, New Zealand, we're going to be bringing China in very soon, are looking at these standards to make sure that these data sets are going to be retrievable and accessible. The NSF data curation mandate that came down back in May, telling us that it was going to happen initially in October, and then was affirmed to be going into place January 18th, 2011, became a real incentive for researchers to look at how are we going to describe our data. At Purdue and in the libraries, and I think on the campus as a whole, we knew this was coming. We knew that this was going to be a requirement from NSF. So we've been working for the last 3 to 4 years identifying how are we going to meet this kind of requirement of data management. And for those of you who aren't familiar, this is just a very brief summary of what the expectations are in the data management plan. They expect the types of data to be defined, the standards to be used for data and metadata, policies for access and sharing, policies for reuse, redistribution, and protection and production of derivatives, and plans for archiving data and the preservation. So once we knew that this was going to come about, Jerry McCartney, the CIO here, and I met with Richard Bacchius, the Vice President for Research, and we said, we need to do something. We need to pull together a group of people to really sit down and look at how are we going to address and respond to the data management plan requirement. We brought together a task force of faculty and from all the different disciplines, from all the different colleges, and Jerry and I co-chaired it. And we worked through a lot of the issues that were going to be confronting us in coming up with the data management plan. And really, the issues were about as many faculty there are on campus. So in preparation and knowing that this was going to happen, the Purdue Libraries, along with the School of Library and Information Science at the University of Illinois, Urbana-Champaign, submitted a grant to a federal funding agency, the Institute for Museum and Library Services, to start studying what are the issues with data curation. How do researchers manage data? How do they use data? Because it became very clear to us very early on, all disciplines, all researchers use data differently. And so we started working on projects. We took certain disciplines, University of Illinois took disciplines. And then we completed our research on this. Then we said, okay, now we need to help librarians understand and how to go out and work with faculty in managing data. And so Purdue got a second grant to go out and start developing a profile and a mechanism for being able to sit down with a faculty member and start describing the data. And this was just released this morning, the Data Curation Profiles Toolkit. Now, this has not been specifically created to answer the questions of the data management plan. We are doing tests now with faculty here at Purdue to see how we can modify this toolkit to specifically answer the questions that a researcher will need to comply with, or need to answer in order to comply with the data management plan at the National Science Foundation. And this is just another page from the data curation profile. And this is the website. So anybody can go on there right now and see it and start. We're going to have a formal release of it in January, but we're making a soft release today. And then as a result of the work that the task force did this summer, ITAP, the libraries, and the Office for, or the Vice President for Research Office, we have been working together, and I think John's going to speak a little bit more about this later, but we're creating a hub that will be a facility for people to house their data. And this is a very collaborative effort between ITAP, the libraries, and the research office, because we're concerned about the content, how the content is described using metadata, just as we would be with any collection in the library, and making sure that that collection is stewarded into the future. ITAP's responsibility will help us to keep that, the infrastructure running, the cyber infrastructure, the hardware. So it's a wonderful collaborative relationship. I think we probably have the best proposal for managing the issue of data management plan in the country. So I'll go stop there. So thank you. I only had 10 minutes.
Hello, everyone. Again, my name is Sorin. I want to acknowledge my colleagues, Lorraine Kisselburgh and Seungyoon Lee, if she is still here. I think she went to, there she is. And also my graduate student, Brian Britt, who's been working with us for the past 2 years. Our umbrella project is called Visible Symbiosis, and it's meant to leverage the Purdue cyber infrastructure for studying large-scale knowledge communities. More specifically, we are in the process of parsing out the Wikipedia data set to figure out if there might be some underlying structure in this data set, and if this underlying structure might be indicative of some transmedia processes, or processes that could be applied to other media environments. This is a massive data set. We're talking about all edits made to Wikipedia between 2001 and 2008. That includes edits made to about 17 million articles, about 280 million edits, and we're talking about 20 million unique editors. Now, by studying Wikipedia, again, we're trying to get at its underlying collaborative structure, and I'll describe that in a minute. But we are also trying to understand how knowledge emerges through collaboration. So this is a two-pronged approach. Let me talk a little bit about the first approach, about the approach that will lead us to understand a little bit better what's going on Wikipedia in terms of social structures of collaboration. This approach is put under this fanciful umbrella title, Decoding the Social Media Genome. And what do I mean by this? Well, if you were to map all the editorial interactions that have occurred on Wikipedia over the past 10 years, you would get something like this, actually something much more complex than that. That's just one article, right? When we are to map all those 280 million edits, you'd get something that this campus would not be enough to cover. How do you make sense of this? How do you approach a problem like this? And we propose that we should approach it using a network analysis perspective, which basically would take every individual that's made a contribution to Wikipedia and connect that individual to other individuals who have made contributions to the same articles. And thus you'd get this gigantic network. And then after you get to this very complex network, then you would start weighing the connections by looking at how many words these individuals have contributed to the project and how far apart the contributions were in time, so that your network gets a little bit sparser. You can filter out the connections that are too weak. And you'd get something in the terabytes of information. And when you got to this point, you have this gigantic network with edges that are weighted and all that. Then you might want to look a little bit more carefully at the embedded subnetworks that make this gigantic network, right? You can imagine an article where, you know, there's an individual who's really central and who works closely with all the other individuals because he or she is there all the time, and, you know, there's like a star-like network in this article. And you might realize that all articles actually have the star-like structure in the beginning, which with time evolves to become a more complex subnetwork, which we don't know what it looks like right now. But by decrypting this first level substructure, in a way we are in a position, we would be in the same position molecular biologists were when they realized that DNA, as complex as that molecule is, could be actually reduced to a number of basic bases, those 4 bases, right? And that, in effect, you know, DNA does its magic by the way in which these bases recombine. So if we were to find these basic substructures, and then if we were to figure out what rules could be applied for divining how they come together to create ever-increasing complex networks, I think that we would be in a better position to understand what is really happening on Wikipedia or on other social media like it that use social network or that allow social network analysis in a way. So in effect, what we're trying to do here, we're trying to parse out a very complex problem into a simpler, more manageable problem. And in order to do this, and I laid out this alphabet soup here, you know, chromosome, structure, network, and all that, just to kind of make it a little bit more understandable, so to speak. If we are to follow the same path, you know, obviously that we would need to deploy very laborious and time-consuming analysis if it were to be run on one computer. So we are trying to leverage the TeraGrid resources and the CodesNode resources for, you know, deploying a massive collaborative effort of defining if there are any basic structures on Wikipedia. Now, let's say that we have solved this problem. We figured out what the local networks are and how they recombine and all that. Now, if we only did that, we would still be in the position of just figuring out some basic structures and just kind of mapping things out. Could there be a more synthetic way at getting at this problem? Could there be a way of telling, by using one indicator, if a network, either local or global, is more or less structured? And for doing this, we think that we could use a social entropy approach by which you basically look at what is the variety of contributions made to a specific network. And if a network is dominated by a few nodes, then the entropy goes down. And if the contributions are even or absolutely equal, then entropy goes up. And just to give you a preview of what we've done so far with the resources generously offered by our infrastructure here. We have looked at Wikipedia as a whole, and we calculated its level of entropy over the course of its existence, trying to figure out at the very first cut if Wikipedia as a whole has become more or less structured, more or less complex, or more or less uneven, if you want. And this is what we found. This is a very interesting finding. And the way you read this graph is like this. The dotted line, black dotted line, indicates the curve for the situation where all articles were edited in a totally equal, even manner at every single point in their lifetime. On the x-axis, you have the number of iterations that articles on Wikipedia could have gone through, and on the y-axis you have the entropy level. So basically, at point 0 you have all articles that started at point 0, and they have a specific level of entropy, which is very, which is at 0 point. And then where everything is even. And then you have the observed editorial activity, which is the orange line, which, as you can see, diverges from the situation of absolute evenness. So basically, Wikipedia has become more and more uneven and more structured, so to speak. Now, this is a very first cut analysis. It uses a very rough methodology that only leverages to a certain extent our resources here, but we intend to redo this analysis using every single article in part and looking at every single article and then combining this into a bigger and more refined story. Now, once we would have been done with this operation of understanding both the basic social networks and also the small global evolution of Wikipedia, we would like to engage in a different type of research that would look at how these structures and how this evolution leads to a specific type of knowledge. Right? What are the dynamic processes that produce knowledge? And here we would look at specific topics and specific ideas and how they create their own meaning knowledge and how these meaning knowledge are connected to social networks. So basically we're talking about a type of research that looks at co-evolution of networks. And this is a unique type of research program in that it looks at massive amounts of data, both in terms of individuals to participate in the social networks, but also in terms of the amount of knowledge that these individuals have generated. And this is just like an illustration of how complex that could be. Now, by way of implication and just to conclude, I think that we have here a unique opportunity to deploy a massive research project that looks at one of the most influential social and knowledge projects of our time. And I think that we have a wonderful opportunity here at Purdue to do this and do it right. And I'm saying it using these big words because I also want to signal that this project is meant to be an open project. What we want to do with the project is not only to generate 3, 4, or 5, or 6 papers, myself and my colleagues, and go after 1 or 2 or 3 grants. What we would like to do is to position Purdue as a center of excellence for studying massive social media data sets. And Wikipedia would be just the beginning. What we would like to do is to bring more data sets and analyze them in conjunction with our colleagues from as many disciplines as possible. As you probably heard, Twitter just released its data set, and it's stored at National Archives or the Library of Congress, and we would actually like to bring it here and have it installed in the same environment, have it analyzed using the same methodologies. We're thinking about bringing the Linux kernel, which is significant not only for its technical capabilities, but also through the fact that it is a social product. So I would like to conclude by saying that this project is actually an open-ended project that hopes to attract more collaborators from across our campus or from the learning community. Thank you.
Yeah, as he said, I am the director of the Purdue Genomics Core Facility, and I'm just going to talk about today one of the services, or one of our major services, is sequencing DNA, and how what the computational requirements for one new type of sequencing DNA called next generation sequencing is. So let's see here. I just realized I was talking earlier that I was kind of jumping the gun on the number of staff I have here. I actually have 9 FTEs. I have 2 part-time staff, students that might contribute to be a 10th person. But we really have 5 lab staff members and 3 informaticians, and we're used by greater than 100 labs on campus, largely for the smaller type of sequencing that we do, smaller data sets. But I'm going to be talking mainly about a larger type of data set we generate. So some of you might be wondering why bother sequencing DNA at all. It's the information storing molecule of most types of life, all types of life, if you don't consider viruses to be alive. It's a molecular polymer for information storage. It's very dense, nanometer scale. And to give you an idea of what that actually might mean, 1 microgram of DNA, a millionth of a gram, is roughly 10 to the 15th bases. And each base has 4 different possibilities, so that's 2 bits. And you work that out, it works out to about 250 terabytes could be encoded and stored in a cubic millimeter of aqueous solution. Well, in principle, how about in reality? Actually, my calculation here is wrong, but anyway, 12 gigabits, I think this part should be correct, are encoded in in every human nucleus, using a human nucleus in this case. A human nucleus is not 7 cubic micrometers, sorry, it's 64, and it's not 18 gigabytes in a cubic millimeter. That would actually come out to be 12 petabytes per cubic millimeter. That's if you packed all the nuclei together. So a better figure here is actually 12 megabytes per cubic micrometer. That's just the data storage capacity of a cell nucleus in a eukaryotic cell, and actually a bacterial cell. So it works out to be exactly the same thing for a bacterium like E. coli. I did the calculation as well. Also, another reason we study, as molecular biologists study DNA, is it has built-in amplifiability. So a lot of the stuff that's going on in biology is happening at a very small scale. At the nanometer scale, it's really too small for us to detect, for the most part, so being able to amplify that signal is really part of the way DNA is designed, and we take advantage of that. So those are the main reasons we use it, we study it. Some other reasons: we can isolate the DNA away from all the other stuff going on inside the cell, the cellular glop, I'm calling it, no disrespect intended. The DNA itself has high stability compared to many of the other structures inside of a cell. Compared to many proteins that are going to be present in the cell, it's actually quite temperature and base and basic pH stable. And so that allows us, once we've isolated it away from all the other stuff, to store it and purify it. And so basically all it comes down to, it's fairly easy to work with. Even though it has a moderately complex structure, it's actually simple compared to the actual cell or organism that it encodes information. Just briefly, this is DNA. This is a cartoon of DNA squashed flat. Just to give you an idea of what's going on, I said it was a nanometer scale. The pitch between these, so the information is stored in the actual identity of these bases here. And so if you split this in half this way, you have information encoded in these different types of bases, the A, C, T, G. And then on the other strand, you actually have another copy of it, but it's stored in a complementary fashion. And then the actual size that we're talking about here is between these different rungs of this ladder. You can see that are attached by these phosphodiester backbone is 0.3, about 0.3 nanometers, so actually sub-nanometer scale. And the diameter of the more or less cylindrical structure is 2.2 nanometers. Okay, so that's a lot of potential information. If you think about 10 to 100 trillion cells in a human, and anything that's about the same size as us, you could scale it different ways, but that's a very large amount of information that's available out there for us as biologists we might want to access. So how do we actually read that information? In the past, we used a method that has subsequently been named the Sanger method. And using that methodology and modern machines, we're able to produce about 1 million bases a day at about $1,200 per million base pairs. Around 2005, 2006, there was a new type of sequencing paradigms began to be employed, represented by 2 of the different platforms that are now available that we happen to have in the Genomics core facility here at Purdue. One type can produce roughly 400 million bases a day at $25 per million base pairs. And here's another one that actually can produce currently around 6 billion bases a day at about 50 cents per million base pair. And that's the second generation, first generation. There's third generation machines that will probably be released this year, and who knows what they'll be able to do. All right, this is up to the point I mentioned for second generation sequencer. This is just to show very quickly that there really is a exponential increase in the amount of sequences and total number of bases that are being produced by scientists, because this is just the database like GenBank that stores all of the submitted sequences, and you can see here that it's increasing in a log linear fashion over time. So who cares, though? As long as my computer is, as Moore's law is happening, as the prices of my computer are dropping fast enough, I don't really care how big the data gets. I'll always have powerful enough computers to be able to deal with the massive amounts of data that's available, right? And Moore's law is still working, so we should be in good shape if that's the case. If Moore's law does predominate, I can spend most of my grant money on reagents and staff. That's basically the way to look at that. So is that the case? Sadly, no. What you're seeing here is basically the curve of the cost per million bases of DNA, per million of bases of DNA for DNA sequence, and then we have plotted on the same graph the decrease in cost of computing, based just kind of general way of stating Moore's law, and see how well they're matched. Actually, they weren't too badly matched until, if you can kind of imagine 2 lines here, one going here that has a slope that's not too much different on this log linear plot again than Moore's law. Then it all kind of it just falls apart around 2005 with the addition of next generation sequencers, and we're in big trouble down the road. At some point, we just are not going to have enough computational capacity to be able to deal with all of this data without spending more money on the computer capacity than we are on reagents, at least if this continues at this rate. So I just want to go into what we actually have in the lab and what we use to process data at the current time. So we have a console equipment in the lab, which includes some small Linux clusters. In one case, the case of the solid, which is the instrument I'm going to focus on for the rest of this talk, it mainly does, it does the primary processing and actually infers the sequence from some image data that's collected. Past that point, once we actually have the sequence data, we use downstream equipment, and we work with Michael Gribsko, biology, and we share servers with him and also nodes on coats to do the downstream processing. And we have two different places that we process the information. One is in Smith Hall. We actually use this after what I'm going to call secondary processing, after we've actually determined the sequence and mapped it back to the genome. Then we need to have a place where we're going to give this information back to the customers so that they can access it, basically websites. And for doing that, but also they may also want to do other, say, tertiary types of analysis. And we have various servers that are available there to do that. This is in Smith Hall. And then the majority of our secondary processing, however, is done on the Coates cluster nodes. And these include 8 16-core nodes, so that'd be 128 cores that we use. And also we have about 7.5 terabytes of Blue Arc storage that we feed the data from. Okay, so how much usage are we currently using? For a solid or an Illumina sequencer, you kind of generate data as a flow cell. The solid actually has 2 flow cells, and during a 2-week period, which is the length of time it takes to do a run, you generate about 50 billion raw bases of sequence currently. And through secondary processing, the amount of, basically the secondary processing involves taking these 50 billion bases and aligning them back to a reference genome and seeing where all of those reads map. And that process, which is generally undertaken, well, actually it's always undertaken by Rick Westerman, one of the informatics specialists at Purdue, at the Genomics core facility. Basically, Rick did this calculation and determined it takes us about 1,000 CPU hours, and that generates close to a terabyte of files.
My name is Jing Xia. I'm a PhD student in the Department of Statistics. Today I'm here to introduce to you a distributed computing environment for data-intensive analysis. Its name is Huipei, and it's developed by my former colleague, Saptarshi Guha. Saptarshi has graduated this summer, and he's working in Revolution Analytics right now to continue developing computation environments for people in need of with large amount of data. First, I want to say a little bit about the motivation. As we all know, data-intensive analysis is commonly challenged in data storage and maybe every computation task. People have to think about how to store the data and in which format. And usually, in order to achieve that, people go to low-level languages. And as a result, people have to spend a lot of time in programming instead of thinking about the problem. And also, if you go to low-level language, when you want to use data visualization tools to help the modeling, you may need to go back to a higher level language. So you have to jump between computing environments, and that's not what we want. So our aim is to build a computing environment that is first exclusive. We want to be able to handle data storage, computation, and visualization in one environment. And also, we want the environment to be easy in programming so that data analysts can save more time in thinking about the problem instead of writing all the detailed implementations. And in the end, we want the environment to be fast in computation so that we don't need to wait for weeks in order to finish a task, so that we have time to try out more models. Before I introduce the Huipei, I need to first introduce to you a programming language called R. R is the most widely used programming language for statistical analysis. Most statisticians use R and SAS for statistical analysis. The research community tend to use R more than SAS, while the application community and the industry tend to use SAS more. R comes from the S language. John Chambers developed the S language while he was in Biolab. And the S language is so pretty that in 1998, it won the ACM Software System Award. It's the award in software system community. And other award winners include like World Wide Web, Linux, and so on. As is commercial, and two professors in Australia developed a free implementation of S and call it R. R has many advantages. First, it's very easy to program. You can avoid a lot of the detailed implementation in R. Programming R is just like issuing the commands in words. And second, R is highly extensible. Users can submit packages by their own to extend the functionality of R. Currently, there are 2,600 additional packages as of yesterday. So you can see how powerful R can be. And also, R provides comprehensive graphical functionality, which is very useful in data analysis. And in the end, R is maintained by a highly active R developer core team. Many people may think that since R is free, it may not be a good language. In fact, it's not. Because of this high active core team and the ability to let users submit packages, R sometimes updates faster than commercial languages. So it's the best programming language for statistical analysis. However, R does have a disadvantage, which is that it cannot handle very large data, which is one of the major reasons for the industry to still using SAS, even though SAS charge them a lot. Okay, and then I need to introduce to you a distributed computing platform called Hadoop. Hadoop is targeting for data-intensive applications. It enables you to work with thousands of nodes and petabytes of data. Hadoop is also free. Google proposed the Google file system and Google's MapReduced computing model in order to achieve distributed computing for their data. And then the developer of Hadoop is inspired by Google's work and decided to create a free implementation so that everyone in the world can use this platform for free. So this platform, Hadoop, is mainly composed of two parts: the Hadoop distributed file system, the HDFS, and the MapReduce distributed computing engine. The HDFS is special in two things. First, it stores large files in small blocks of certain size, so that every time only a small block of data is processed, this is used to help achieve computing efficiency. And second, HDFS creates multiple copies of data across different machines. So on one hand, when a part of data needs to be processed, the HDFS finds the copy that's closest to the processor. This is also used to achieve efficiency. And on the other hand, this is to ensure the safety of the data. If one copy is lost, we still have two copies to use. The MapReduce distributed computing engine performs analysis on the blocks of data that's saved in HDFS through map and reduce steps. And I will explain about the two steps in the next slide. Hadoop is greatly sponsored by Yahoo, and is also used by Amazon, EC2 and S3 computing services, and IBM, and many other big companies that have large data. So it's now a most popular distributed computing platform that has been used by the world. And Purdue has launched its Hadoop Cluster 2 earlier this year. Okay. Now I just want to explain a little bit about the MapReduce computing engine, just to make the story complete. So first, in this engine, we save data in the format of a key-value pair. This is similar to what people usually do in creating a data object. Key is like the name of the object, and value is the value of the object. The only difference is that the keys can be replicated. The map step performs user-defined operations on all data in parallel. This is also similar to what people usually do in data analysis. The only difference is that these operations are carried out in parallel across the cluster. The reduce step is a little bit different. Reduce is not necessary, but it is useful. It can be very useful sometimes. In the reduce step, if you want to carry out operation on data that have the same key from the MapReduce, in other words, you want to aggregate some data, then reduce performs the aggregations for you, and then you can carry out the operations that you want on this aggregated data. Okay, so HurryPay is an integration of R and Hadoop, the two computing environments I've just introduced. HurryPay is called, the full name is R and Hadoop Integrated Processing Environment, and it's developed by Purdue graduate Saptarshi Guha this year. The strength of HurryPay is that it provides data analysts a comprehensive environment completely in R. You can do the data storage, the computation, and visualization completely in R, and you don't need to worry about Hadoop. You don't need to learn about it. And also not just a combination. It inherits most of the strengths of R and Hadoop, and as a result, it makes data-intensive distribution analysis extremely easy and highly efficient. Since R and Hadoop are both free, HurryPay is free too. Here I list a couple of specific features, but I guess I'm going to skip that. I do want to mention HurryPay. It's Greek, and it means a moment in time. Okay, here I just want to briefly show you the programming structure of HurryPay. You can see on the top, the users define a map process in an expression. And then in the middle, the user define a reduce process also as an expression. The reduce process includes three parts. In the pre-part, you basically do some setup operations like initializing some variables. And in the reduce, you get output from map, and then you can carry out the operations. And in the post, users can do some write-up operations. And then after you define the map and reduce process steps, you initialize this map reduce process by this function called RHMR. And after you initialize this function, you can execute it by RHEX. So basically, MapReduce process can be carried out by just writing these 4 steps. Here is an example. I just want to show you that with this these less than 30 lines of commands, I'm able to compute. Compute a regression model for each object, and for the objects that belongs to the same site, I aggregate them and take an average of their regression fits. We perform this regression on 3.7 million objects, and that in total there are 885 million observations, and the total size of the data is almost 14 gigabytes. But we're able to achieve this in 96 seconds. Okay, in the end, I just want to briefly introduce our cluster. Our current cluster have 8 servers and 78 processors in total. There are 400 gigabytes memory and 10 terabytes disk space. And the former example that I just gave you is performed on this cluster. And we're going to have a new cluster, and this is in collaboration with ATAP. With this new cluster, we're going to have 11 servers with 264 processors, which is going to greatly expand our capability in fast computation. And we have 500 gigabytes memory and 59 terabytes of disk space. And the total cost of these 11 servers is only $50,000, which is pretty cheap comparing to other expenses that might be involved in data-intensive analysis. This cluster is going to be managed, or is currently under management by ATAP. And with this cluster, first we open it to the research group in the Department of Statistics, and we want to use this as an experiment to see how Hadoop, whether Huipei can help a large amount of users doing data-intensive analysis, and maybe this will become a transition to a larger cluster that can serve the Purdue research community. Thank you.
I know that we've run over time, and I know that that's a consistent trend. I think I just talked with John, and he said to ask for a few questions if we have them. So if anyone in the audience has questions for any of the panelists, please request the microphone, and then direct your question either towards all of the speakers or any specific speaker, and we can have a few moments of conversation about the topic of this panel discussion before we move on to the next event. We have a hand up in the far back.
Hi, I have a question regarding the library speaker that was here earlier, Dr. Mullins, I believe it was. You were talking about the derivatives of data sets that we left behind. One of the things I'm interested in is like this a concept of a chain of custody, or how a data set evolves as computation is applied to it. Is there anything in your current scheme that includes that?
Yes. If you're talking about how to do the site, is there a microphone?
Get my phone just up.
Are you referring to being able to define or to cite the derivatives or see the chain, the lineage of the data set?
More or less, yes.
Yeah. The DOI is going to be buildable. The DOI that you would assign to an initial data set can then have add-ons to it, so that as the data set generates into or is regenerated into different data sets, that you could create a DOI that would show the lineage of the relationship. There's a colleague that we have at Los Alamos Research Laboratories, Herbert van de Sampel, who is dealing with exactly this issue of trying to determine how are we going to illustrate and how are we going to document the lineage of a data set. And so there is great concern about that.